knew Karen, and she knew that Karen had given birth to the children. The chances that not just one but two of Karen's sons had been mistakenly switched at birth was also astronomically unlikely. So too was the chance that there had been a mix-up in the blood lab. On a hunch, Uhl decided to check Karen's blood sample against some tissue from another part of Karen's body. This test solved the riddle. Karen did not have one DNA sequence or genome in her cells. She had two. 53 years before, early in Karen's mother's pregnancy, two separate eggs had been independently fertilized, giving rise to two separate balls of cells, each with its own DNA. At some point, in the rush of cell division and multiplication that follows fertilization of the egg by sperm, the two groups of cells fused into one. Instead of developing into twins, they developed into Karen, with cells from both balls randomly distributed throughout her one body. While most of Karen's body had cells from one of the groups, it just so happened that two of her sons came from eggs that had been generated by the other. People who carry more than one complete genome are called chimeras, after the fire-breathing lion of Greek mythology, with the head of a goat growing out of its back and the head of a snake growing out of its tail. The term denotes that they are combinations of more than one creature. Karen is not alone in being a natural chimera. Indeed, the first human chimera was identified in 1953, the same year that the double helix structure of DNA was discovered. And today, some scientists estimate that about 15% of people are chimeras. Sometimes only blood cells are mixed up, but other times, as in Karen's case, two separately fertilized eggs start to develop and then get fused together. Ever since the day in 1953 when James Watson and Francis Crick unveiled their model of the double helix to account for the structure of DNA, we've been in thrall to genes. We think of every aspect of ourselves as being determined by our DNA, from the colour of our eyes to our propensity for a particular disease. In the minds of some, DNA even sets the parameters for a person's intellectual ability or temperament. It's in her genes, a parent will say about a child. We take a swab of cells from the cheek and get our DNA tested to learn who we are, as if tracing which genes we inherited from whom tells us anything about ourselves right now. DNA has become so central to our sense of identity that we even use it as a metaphor for social organizations. It's in our DNA as a company, a CEO will say, or as a team, says a coach. Yet chimeras are just one way in which nature shows us that DNA does not define who we are. Karen is not defined by a DNA sequence. She has two. The publication of the human genome ushered in an era in which people think that most non-infectious diseases have some genetic basis, underscoring the connection between DNA and us. For conditions linked to a mistake in a single gene, like cystic fibrosis, haemophilia, or sickle cell anemia, a focus on DNA will almost certainly allow scientists to develop cures. Along these lines recently, cutting-edge technologies like CRISPR, the so-called genetic scissors that allow editing of DNA at will, have thrown up a host of potential treatments. For example, gene editing interventions using CRISPR, an acronym for clustered, regularly interspaced, short, palindromic repeats, have been shown to repair a single change in the DNA for the beta-globin gene, HBB, that produces sickle cell anemia and thereby restore the health of individuals. Other cases are in the pipeline. But even in instances like this, there are problems. The relationship between changes in a gene and a dysfunction is not usually as straightforward as in the case of sickle cell anemia. Having mutations in the breast cancer type 1, BRCA1, or type 2, BRCA2 gene makes it more likely that the body can't produce the functional proteins needed to effectively destroy cancer cells in breast tissue, but it does not say you will get cancer. Mapping gene mutations to cell malfunctions may help us to understand what happens when a gene is faulty or absent, but more often than you think, the observation doesn't tell us how cells use the normal form of the gene to make normal tissues and organs. In fact, over 60% of birth abnormalities cannot be linked to to specific.
To keep listening to this audiobook, I've left you a link in the description so you can do it at no cost thanks to Audible's 30-day free trial.